Good morning to all. I am Dr. Tikamchand Dakal, presently working as an assistant professor in MLS University, Udipur, Rajasthan. Slide 2. We are passing through one of the toughest phases of our lives. It's a critical time for all of us and our community to act smartly and spread the awareness for the prevention of the disease. Coronavirus is a highly contagious virus that causes infection to human respiratory system by binding to lung epithelial cells. Coronavirus have spike proteins on their surface through which they attach to the host cells and gain entry into them. The infectivity of 2019 novel coronavirus is unimaginably higher than all previous known coronaviruses, and the reasons are largely unknown till today. Coronavirus has been rapidly infecting people around the world, which was first detected when a cluster of mysterious pneumonia cases emerged in China's Wuhan city late last year. It has various proteins that help them invade human cells. One of the important structures is the spike protein, which helps in the attachment of the virus with the cell receptors. During infection, the S protein is cleaved into the N-terminal S1 subunit and C-terminal S2 subunit by host proteases. S1 consists of the N-terminal domain (NTD) and the receptor binding domain (RBD), is critical in determining tissue tropism and host ranges. The RBD is responsible for binding to the cell receptor, which makes the virus to hijack the host mechanism and replicate into millions of copies to infect the healthy human cells. Various phytochemicals have been identified by computational drug development approaches to be effective against SARS-CoV-2. In our study, we have taken 100 potential phytochemicals that have been studied extensively in various antiviral interactions with coronavirus, and analyzed their interactions with spike protein by using in-silico approaches. Slide 3. In our study, 100 phytochemicals with reported antiviral properties were selected from literature. These were then subjected to various physiochemical and pharmacokinetic analyses, and possible targets were predicted. Tools used for predicting the drug likeliness of the phytochemicals were SwissADM, PASS, Swiss Target Prediction, DIGPRED. The phytochemicals fulfilling all the criteria were then filtered out and subjected to molecular docking with the receptor binding domain of the spike protein, and was analyzed with UCSF Chimera. Slide 4. Phytochemical compounds were analyzed using SwissADM based on physiochemical and pharmacokinetics properties. Physiochemical properties such as number of aromatic heavy atoms, fraction CsP3, rotatable bonds, H-bond receptors and donors, molecular refractivity, lipophilicity and water solubility was determined. Pharmacokinetics properties included GI absorption, blood-brain barrier (BBB), permeation, permeability, glycoprotein (PGP), and drug metabolism. As per RO5, Lipinski's rule of 5, a chemical compound to be orally active in human should follow minimum 3 criteria of the following: A, molecular weight 500, B, XLOGP3 less than 3.5, C, hydrogen bond acceptor 10 and hydrogen bond donor 5. Therefore, compounds following the rules are noted and others are knocked out. An example of few compounds is given in the table. Slide 5. Next analysis was done by predicting biological activity using PASS. It evaluates general biological potential for drug-like compounds based on their structural formula. Possible biological activity of all the compounds retrieved were analyzed, and the properties could involve in the disease regulation is taken into consideration. Properties such as anti-inflammatory, antioxidant, HIV-1 integrase, antioxidant were taken into consideration. Slide 6. Following PASS, Swiss target prediction was used for predicting the biological targets of the drugs. In the current study, we observed many compounds showing structural similarity and active biological target molecules in the human species. Molecules with higher probability has been selected. Slide 7. Later on, C was used for predicting the biological targets of the drugs. Slide 8. To conclude, gene expression studies of the compounds was also evaluated using DIGPRED. That showed a plethora of genes involved in the various kinds of biological activities that has been regulated either positively or negatively by the compounds. In our study we have taken 5 genes, namely PRDX2, CD14, CD86, CD83, CCL3 and CCL2 are taken into account from the displayed genes as they could be directly involved in the immunogenicity of the virus and cytokine regulation during disease. Slide 9. The compounds fulfilling all the characteristics of drug likeliness was then selected for molecular study. Out of 100 phytochemical compounds, 20 compounds were found to get passed in the analysis. These compounds were then subjected to molecular docking using Swiss dock. Although almost all of them showed interaction with the spike protein, in our study we have selected 2 phytochemicals that showed high interactions at the receptor binding domain of the spike protein. This slide shows our first compound, apigenin, interacting with the RBD domain. It interacts at 3 positions in the RBD domain, forming hydrogen bond interactions. Slide 10. Our next compound is orientin, which interacts with RBD through single hydrogen bond formation and double hydrogen bond formation. Slide 11. Our study demonstrated that the antiviral compounds screened in the study can be grouped in 2 categories. 1. Compounds that block SARS-CoV-2 interaction with host cell ACE2 receptors and prevent infection, multiplication and pathogenesis, and 2. Compounds that could modulate and boost the immune system of the diseased patients to avoid pathophysiological outcomes, such as cytokine storm. Compounds of former category hold immense potential in the development of herbal medicine against SARS-CoV-2. While compounds of latter category can be used for preparation of powered medicinal formulation, such as Kedha, an Indian Ayurvedic natural immunity booster, we believe that our study could prove be a robust therapeutic solution for treatment of COVID-19 patients using plant-derived antiviral phytochemicals compounds and the medicinal formulations made from them. Slide 12. This study is a novel effort to identify the phytochemicals that hold drug-like properties and also interacts with the viral protein. We have filtered the 100 phytochemical compounds through various criteria to make sure that the compound holds efficient drug-like properties inside our body.